And with that, let me move along to Dr. Todd Kuiken from the Woodrow Wilson Center just down the road, Ecological Risks and Benefits. Todd?
So I want to thank the committee for allowing me to come to talk to you today. I apologize, I'm a bit of a cold, so if I'm a little foggy, that's what that is. So I'm an environmental scientist by training, so I usually get to play the role of Chicken Little there, but with gene drives, everyone else has sort of stolen our thunder with that in a bit, I feel. And so what my goal is today is to raise some of the research gaps that we've identified in terms of potentially being able to evaluate the ecological implications, both positive and negative, of gene drives, and to kind of go through the landscape of the synthetic biology community with gene drives. I would sort of include in that a bit about an NSF grant that we had that developed a research agenda to be able to address some of these issues, and then some potential applications with that. And then I didn't include this in my slides, but it came up a couple of times in the previous session about some of the international issues in this, and I've been involved in the negotiations at the UN Convention on Biological Diversity, where synthetic biology and gene drives is currently being discussed about a new potential framework of how to deal with that. So we can talk about that later if you want. So this is a map of what we have found as sort of the current activity around synthetic biology in the globe. And as we heard previously from Kevin, sort of talking about sort of the accessibility of this technology, I think it's important to realize that this is not just a US-focused potential application that can be used. And more importantly, I think, is this map, which is the iGEM map for this year's teams that are currently registered. So iGEM is the International Genetically Engineered Machines Competition. It happens every year. It started as an undergraduate research competition for synthetic biology. It started at MIT with, I think, 5 teams, and there were 50 parts, sort of the genetic parts that were entered into the registry. This year we have over 280 teams from every continent in the globe, including high school teams and community laboratories. So if we sort of believe sort of what we're hearing about sort of the development of this technology and it's that accessible, I think we need to think about this in a much broader scope about who's going to have access to this technology and what types of systems we need to have in place so they can potentially utilize that technology safely. On top of this, iGEM also now enables community laboratories to enter, which is another aspect that I think this committee should at least think about as they're deliberating on these things, that we're not just potentially talking about science being done in academic labs or in university labs or professional labs in industry, but we now have this movement of the DIY bio community where this is GenSpace, which was the first community lab which opened in Brooklyn in 2011. But rapidly we have been seeing more and more of these open across the globe. I'm not saying that they have the capabilities to do this type of sophisticated work today, but I think it would be prudent for the community to think about that there may be a potential for these types of labs to also have access to this technology, particularly if it's under $10,000, which Kevin had mentioned before. This is some new data that we're still tweaking, so some of these numbers may change, but this is the current US funding of synthetic biology since 2008. And since 2008, there's been about $580 million, from what we can tell, has been invested in synthetic biology. But if you look at sort of where a lot of these numbers are coming from, that orange box should really sort of stand out to you. So that's the DARPA funding. Now that's the public funding that we know about. And why I bring this up is because of sort of some of the agencies that are missing here, particularly when we're talking about the ecological implications. EPA is not anywhere on this graph. They have invested $0 in terms of investigating synthetic biology research. But in particular, so we tried to pull out all of the ELSI funding that we could find within this, and we could only find about $9 million since 2008, or 2006, actually. I expanded that out. But if you even separate that out even further, the risk research is only about $6 million, which is about 1% of the total of that. And again, if you look at the agencies that are missing, so EPA isn't on here, Fish and Wildlife Service isn't on here, USGS isn't on here, the agencies that would really have sort of the most expertise to be able to evaluate some of these things currently have no research programs to address those issues. So if you look specifically at some of the DARPA programs, I won't go into this into detail, but they have their Living Foundries program, the BRICS program, which I think is maybe even more pertinent for this discussion, which just launched this year, is where they are actually developing or trying to develop organisms that can survive in complex settings. So they know they're going to try, they're looking for organisms that can survive in essence in any kind of system that you can think about. So most of the organisms now are in controlled laboratory environments that don't do very well outside of those prescribed areas. So they're now investing in areas to be able to develop more robust organisms. There's a new program that's sort of out there, isn't funding yet, looking at synthetic ecosystems, as well as a new program specifically on malaria that would most likely involve gene drives. So this report, and I have hard copies of this because I don't know if this was distributed to the committee, but this was an NSF grant that we received a couple of years ago that Ken and I worked on together, where we were sort of tasked with developing what the research agenda would look like if you were going to evaluate synthetic organisms that were deliberately being released into the environment. And as part of this, we brought together ecologists, evolutionary biologists, as well as synthetic biologists, including Kevin, who actually presented on gene drives to this group, and came to a consensus about some topic areas that we thought NSF in particular should maybe put some money behind to start addressing some of these issues. But just to go over this sort of in some broad scopes, sort of two sort of main things popped out at us amongst those discussions. One was the novelty and the speed of the technology, gene drives in particular, of how fast this technology was moving and the number of changes within that technology along with that speed. The second was this notion of eco-evolutionary dynamics. So how do you evaluate an organism that itself is evolving within an ecosystem that's evolving sort of simultaneously with that organism? They were areas of research that we couldn't identify things where we have enough data to be able to make those determinations in places where we thought more work was needed. The other was this radically different time frame. So if you listen to Kevin and others in the field about how quickly this technology is developing, and some of the comments before that it's inevitable, which I have some concerns about. But that being said, how do we design ecological research programs which tend to go at a much slower pace to try to keep up with the sort of the fastness or of how fast those technologies are actually developing. So these were the 7 priority research areas that our group identified. These are not in any particular order. And I'm not going to go into detail on these because they're explained pretty well in the report we had. But basically sort of comparators, the phenotypic characterization of the organisms, the fitness, genetic stability, and lateral gene transfer, control of organismal traits, this monitoring surveillance, which I think is actually a critical component of this to be able to find where these gene drives are actually moving towards and what they're actually doing. Whether you could develop new modeling techniques to be able to model sort of the interactions that are going to be happening, and then whether you need this standardization of methods and data. I just want to sort of call this out specifically because if the committee is not aware, NIST actually just started a program looking at synthetic biology and to start developing or start thinking about what types of standards that maybe need to be developed for that community. So it's a group that you guys might want to reach out to hear about what they're working on. So we sort of identified all these research areas, and one of the sort of things that I think is missing, one of these gaps, is where you would actually do this type of ecological research if you were going to do it. And I want to call your attention to the Experimental Lakes area, which is a research station in Canada, which is actually set aside to do these large-scale ecological risk studies. I participated in a study in the late 90s and early 2000s where we were able to manipulate. So it's basically a series of it's a couple thousand hectares. I forget how many total lakes there are. But this was the lake that I worked on, looking at mercury cycling. But this particular research station allows you to do the types of manipulations that I think will be needed if you're truly going to try to evaluate the impact of a gene drive, not just on the particular species that you're working on, but all of the subsequent interactions that that species is going to have in an area. And I'm not aware of any type of similar type of station in the US where you could conduct this type of large-scale research. So just to sort of go through some potential sort of hypothetical activities from a positive stance. So we heard, I forget who mentioned about some areas in conservation biology where you might be able to utilize gene drives. And one area that we looked at for the Great Lakes Protection Fund was Asian carp, which I think most people are probably familiar with. It's a huge problem we're having. So this is a map that sort of shows sort of the increase in prevalence of carp over the last 10 years or so. And so we were looking at whether you potentially could utilize a technology like gene drive to go after or to eliminate an invasive species like this. And part of that project not only was looking at the potential of the technology, but also some of the negative consequences of that. And one thing I think in particular that we need to keep in mind is if the technology is successful, whether or not we actually know what happens if we remove an entire species from a system. While this is an invasive species, it's also an established species now. And I don't think we have a good understanding of how to evaluate what happens if we then all of a sudden remove that species from a system as large as this. And just to show you that there are other agencies within the government that are sort of thinking about this, where this wasn't necessarily attached to gene drives. They were part of this project that we did, the USGS, but they are also looking at similar sort of biological controls to deal with some of these problems. Another one that we've sort of been thinking about is this problem of the pythons in the Florida Everglades, and whether something like gene drives could potentially be utilized to deal with this problem. So a recent study that we came across that was doing counts basically of the other wildlife species that are in the Everglades since the pythons were identified, which were some pretty striking numbers. So they did observations from 2003 to 2011. They saw a 99% decrease in the number of raccoons in the Everglades, a 99% decrease in possum, an 87% decrease in bobcat, and they actually failed to observe any rabbits. So this is another area where I think gene drives may have a potential, but again, we really need to be able to understand what all of those interactions would be if we're going to release a gene drive into a system like the Everglades. Whether those gene drives are going to transfer into other species, again, what happens when you remove the species that was the initial of concern, and none of that, which I think right now we have any real clear understanding of how those interactions are working. And more importantly, there's no clear research agenda within the US government to even begin to start addressing those questions. And finally, sort of from a hypothetical to sort of a real life example, so most people are probably familiar with Oxitec and their desire to do field trials of their mosquito in the Florida Keys. I bring this up because it's actually a perfect example of how not to garner public acceptance around a new technology in a particular area. So this is the island where they're planning, where they would like to do this field trial. And just quickly, here are some sort of examples. So one of the issues is they're basically targeting a disease that is not a current problem in the affected area. They're talking about releasing a genetically modified mosquito that most people believe will bite them, even though it's the females of this particular species are the ones that bite. They're releasing the male species, but in essence, that distinction doesn't matter in the public's mind. There's only 440 houses in the area where they're doing this, except this has garnered almost 150,000 signatures against the technology and over 1,600 emails to the guy who runs the mosquito control district in the Florida Keys. And just some quick, sort of real quick data. We just did a national poll asking about gene drives. There's only about a 20% positive feeling in the public about it. There's only about 18% negative, but this number actually is really important, this 62%, because it actually shows that there's space there to have a public dialogue about these technologies and whether or not the public is willing to accept them, whether they want them, and what their concerns and hopes of those would be. We asked the moratorium question. This was on the human side, but my guess is that these numbers would probably hold if you asked about some of the other applications. And this is pretty important. There's a pretty strong support of a moratorium right now until we can figure out some of the issues that this committee is tasked with. So just quickly, the takeaways. I think the U.S. needs to establish an ecological risk research station to do these types of large ecological studies, particularly around gene drives. There definitely needs to be an established, coordinated research agenda that, in my opinion, is co-funding this ecological risk research with the development of the technology simultaneously. I believe DARPA should be encouraged, if not required, to fund this ecological risk research, since from our data, they are the primary funders of synthetic biology right now. I think the committee should think about integrating your findings with the recent White House memo that came out a couple weeks ago, which charged the agencies to take another look at the coordinated framework and their authorities under that of how to regulate these technologies. And then how do we create this culture of responsibility to fill this ecological risk research void that I believe there is? So whether we can do that through the publication culture, through the funding agencies for them developing a culture of responsibility, and also how do we sort of embed that culture within the individual investigators. So with that, I'll stop. I have a list of resources here, and I'll look forward to the discussion.
Great. Thank you, Todd. I appreciate it. All right. What we will do now is move on